Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Esther Walton. Our main subject today is going to be neuroimaging and epigenetics. We're going to talk about Dr. Walton's background and why she chose to study the brain. We're also going to chat about what DNA methylation measured in blood can tell us about the brain and how strongly mental health is linked to physical health and what role epigenetics plays in this association. We'll be talking about a specific measurement Dr. Walton has studied called the brain age and how that is created and how it is associated with epigenetic aging. We'll also dive into developmental timing, what she means by saying aging starts at birth, and why it's important to look at the relationship between methylation, early life stress, the brain, and mental health, and certain associations that appear and disappear at different life stages. A quick introduction into my guest. Dr. Walton's research focuses on the links between early life stress, epigenetics, brain structure, and mental health. As an associate professor in clinical psychology at the University of Bath in the UK, her research on health traits includes depression, ADHD, schizophrenia, and eating disorders, and often consider these in the context of neuroimaging and physical health comorbidities, such as faster aging and cardiovascular diseases. She has hands-on experience leading large-scale collaborations and has also co-established the International Methylation, Imaging, and Neurodevelopment, or MIND Consortium, to shed light on the relationship between epigenetic patterns and brain structure across child development. We'll talk about her consortium today as well. She is an active member and the lead on several projects within the International Enhancing Imaging Genetics through meta-analysis, the Enigma Consortium, and the largest consortium to understand brain structure, function, and disease based on brain imaging and epigenetic data. And now for my guest, Dr. Esther Walton. Welcome to the Everything Epigenetics podcast, Dr. Walton. I'm excited to have you today.
Glad to be here.
Yeah, so you're really an expert on epigenetics in the brain, and really excited to dig into this a little bit further. I know that's going to be the main focus of our chat today, but I'm excited to have you on in particular because little is known about the extent to which DNA methylation is linked to individual differences in the actual brain itself. And I think a lot of the listeners will be ready to really dive into this as well. So, how these associations may unfold over time across development. And there's a time of life when many of these disorders emerge within the brain itself. So before we actually start to talk about that further and unfold that, why the brain? Why are you studying it out of all kind of organs? It's a very complex one. What started your journey into this amazing field?
Well, you're right. The brain is amazing. My journey, well, I suppose it's as often happens, a little bit more complex and more in a roundabout way. So I was in a way always interested in the brain, more from a neuroscience perspective than from a psychological one. But as probably my schoolmates and my teachers can attest, I wasn't the most motivated student in biology in high school. I was always sitting in the back a little. And when it came to going to university, I was a bit undecided. I thought about, maybe medicine or maybe psychology, but also I thought about going into languages or maybe music. So I ended up I signed up to this broad undergrad program in the Netherlands where you could take all sorts of different courses. So there was a course on history, and of course, a few courses on psychology, but also biochemistry and criminology. And it wasn't until the end of those undergrad studies that I thought, well, actually, medical neuroscience is really cool. This is fantastic. And then I got onto a master program in Germany on medical neuroscience, but I didn't get into neuroimaging until the end of that program. And then I didn't get into imaging genetics until my PhD, and only at the end of my PhD did I start working on epigenetic data. So it really took a very long time to get into that area where I'm working now. But just to answer your question about the brain, the brain, in a way, is at the same time amazingly complex, but also not as complex as it should be. So, for example, there is this urban myth that there are as many neurons in the brain as stars in the Milky Way, for example, and we now know that's not true. There are just over 80 billion neurons in the brain, which is still pretty impressive, but it's not as much as stars in the Milky Way. And there's a lot already we know about the brain. We know how neurons communicate. We know how the brain structure works and develops. And we have mapped different brain regions. But what we don't understand still is the link between, the structure of the brain, the stuff we can look at, the stuff we can measure, and how that translates into our personalities, our mind, our mental well-being. And when it comes to the brain, I suppose I, in my research, focus on two things in particular. The one being like, is the brain really the standalone organ for our personality and our mental well-being? So, for example, if you, through some kind of weird sci-fi setup or something, we're able to take the brain out of the body and attach it to some kind of, weird device, would you still have the person there, with all their quirks, all their jokes and memories? Or do you need the periphery for that, right? So we know there's, for example, that, gut-brain axis, and there are, some ideas that the gut influences our mental health and our personality. So do we really need that? Is there a link between physical and mental health? And the other thing that interests me is really what shapes the brain. So we've been involved in a range of studies describing the genetics of the brain. And we found that genetics generally explains roughly around 30% of brain morphology, but that leaves a lot of space for environmental influences and gene-environment interactions. And that's really, where epigenetics comes in. So how does epigenetics relate to the brain itself?
Yeah. Yeah, that's such an interesting story. Thank you for sharing, Dr. Walton. I can see where your passion lies, and I think it's very promising for listeners out there. You were interested in some of the more, maybe psychology a little bit during your undergrad, not really, maybe a little bit of an interest, and then you keep growing that and growing that and growing that. I think especially here in the States, we always feel rushed to choose one thing and then make that one thing our life. But it's beautiful that you're able to grow that through the work you've done in this field so far. And I also appreciate you making something that still is very much complex, the brain itself, but making it much more easy for users to understand that, say, wait a second, it's actually simple. And we're starting to learn more and more about that. So I know I have my list of questions here, but that introduction just made me want to ask, a lot of others, especially highlighting those two others. Is it only the brain where we're getting a lot of these personality traits, or, like those quirks you mentioned, and then what actually shapes the brain? I think one of the major questions I get from people the most is, hey, what part of the body is genetic and, definite, and then what part can we actually change? So that statistic that you also gave, hey, 30% of our brain is probably explained by some genetic factor. It gives us 70% to discover, to understand what epigenetics is doing to the brain. So let's really set the stage for our listeners here. Can you explain what epigenetics and, the DNA modification, DNA methylation in particular, measured in blood, can actually tell us the brain? What do we know so far about that 70%?
Mm-hmm. Well, really good question. So it's quite a contested topic about, can DNA methylation in peripheral tissues such as the blood really tell us something about the brain? And, I certainly had a few heated debates on that. And I think at first the message probably is always not much. Generally the first impression is always that you can't make inferences from blood-based methylation on the brain. However, I come to that but in a second. But first, so why does it look so difficult at the beginning? The one thing being that DNA methylation is actually quite tissue and cell type specific. So if you gave me a methylation data set and you didn't tell me where it came from, I could probably quite quickly tell you, you got that from a brain sample or from a blood sample. And, cell identity is really fundamental to DNA methylation data. So initially you would think, well, yeah, that makes sense. So, making inferences from one tissue to another is probably a little bit more complex.
Mm-hmm.
And to describe that better, we did a study quite a while back already in 2016, where we looked at the correlation between blood-based methylation and brain-based methylation. And there have been people who've done that before we did, but what they usually did is they looked at post-mortem brain tissue, and that comes usually with its own set of limitations. So often in those studies, blood was measured pre-mortem, and then when people died and they donated their brains to science, you could measure DNA methylation signatures in post-mortem brain tissue, but those signatures might decay a little bit or might change with the post-mortem interval because it's fundamentally dead tissue. So what we did differently in our study is that we looked at live brain tissue, and that's exceptionally difficult to get hold of. So we got that tissue from epilepsy patients who were undergoing brain surgery to remove the part of their brain that is causing those epileptic seizures. And you might say, well, that's a huge limitation, right? Because you're looking pretty much at diseased tissue, but we made sure that we looked at DNA methylation profiles obtained from the outer edges of those brain tissues. Gotcha. To get tissue as healthy enough as possible. And we obtained blood samples at the same time from those patients, and then we correlated those epigenetic markers. Now, what we found at the time was quite devastating, and we found that actually only 8% of methylation sites correlated significantly across blood and brain tissue. And they could have suggested, well, actually, we can't really use blood tissue to make any inferences about the brain. But, and now my famous, but I think we can. It works really well, but we have to really honor the complexity of those associations. So, for example, what we find is that there's a time delay at which associations emerge across tissues. Maybe to give you an example, this is completely unrelated, but to help understand this better,
yes, please.
I'm really bad at gardening and at watering my plants, even in the office. Okay. And I never really figured out how to do it properly. And the problem I'm having with watering my plants is that I might forget today to water my plants, but I don't get immediate feedback. My plant doesn't start wilting today or doesn't start dying today. It starts dying in 2 weeks or maybe a month's time. But by that point in time, I can't quite remember what I did 2 weeks ago or something. Did I overwater my plants? Did I underwater my plants? So if I just looked at a cross-sectional association between my watering skills and habits and my plant health, I probably wouldn't find much. Mm. And the same applies to epigenetic data across tissues. So it's fair to assume that, that signal, there's a signal that does transmit across tissues, but it takes time. And we have to model that time delay in a better way. And we have to understand that much better. And there's evidence for, that those cross-tissue signals should be linked, because we know there's a strong link between physical and mental health. And we know that a range of physical health problems, such as increased inflammation, is linked to, depression and schizophrenia. Yeah. And we also know that there's a metabolic component to eating disorders. So we know there's a link. We just have to find better ways of modeling it.
Yeah. I love that example that you gave there. I have a ton of plants at home, and hey, increase in green space actually has some really good positive epigenetic outcomes as well, right? There's a couple of studies out there. So, I mean, you broke that down perfectly. And I think, only time will tell. And again, like you said, only 8% of those DNA methylation markers actually correlate across blood and brain tissue. So we're working with a very limited data set as well. So I think it'll be interesting to look further at some of that post-mortem brain tissue, or even more, I guess you called it more of that live brain tissue. I was actually just speaking with someone from Emory University outside of Atlanta, Georgia, and they have a really good brain bank along with University of Kentucky here, because we're doing a lot of work in Alzheimer's. So to be able to have some of those banks, but again, needing that live tissue is very much necessary. So I appreciate you highlighting that work in 2016 that you performed. My next question, you led me directly into that. You mentioned it very briefly, but if you want to add anything else, Dr. Walton, please feel free. Really, how strongly is mental health linked to physical health itself? And then what role do we actually see epigenetics playing in that association?
Sure. Well, mental health and physical health can be linked quite strongly, and
Okay.
in combination, they can have quite severe health outcomes.
Mm-hmm.
So, for example, there are like excellent studies coming out from Denmark, out of all places. They have like this amazing data set up where they have, access to longitudinal health data from people across the lifespan, or, across at least, 10 or 20 years. And I think like across the whole population of Denmark. So those are really large data sets where you can really describe the link between physical and mental health, and how it leads to, how it impacts mortality, for example. And what those studies have shown is that people with mental health problems, for example, have a reduced life expectancy of 4 years. I mean, that was one particular study. That estimate differs depending on what data you look at. And people with physical health problems have a reduced life expectancy of 6 years. However, people with both physical and mental health problems have a reduced life expectancy of 11 years. So it's quite a step change, from 4 to 6 to all the way to 11 years, suggesting that, the intersection of physical and mental health problems can have huge impacts on well-being in general.
Mm-hmm.
So what we've done in our research is to study the genetic and environmental predictors for physical mental health comorbidity. So, for example, we did a genetic study looking at genetic markers predicting physical mental health comorbidity. So we looked at people who were ill with heart disease, diabetes, and depression. And we found this really strong genetic signal on chromosome 8, and that replicated really well. And that was one study. And then on the other study, we looked at the reverse, like, can we identify environmental risk predictors for multimorbidity? And here we worked with, collaborators. And so we are part of this early cause consortium that's like a cross-European consortium where we pool really large data sets across Europe to really study this across the life course. And what we found, for example, is that a history of childhood maltreatment is a strong predictor for physical mental health comorbidity, much more so than just physical or just like poor physical or just poor mental health. So it's really causing or potentially causing physical mental health problems in combination. And now the logical next step, right, after having found genetic predictors and environmental predictors, is to look like what are the biological pathways mediating this. And we have identified a range of biological pathways, both in the physical space, such as increased BMI, cortisol levels, insulin dysfunction, as well as a few mental health behaviors like sleep problems, for example. And what we're doing now is to see if epigenetics could be one of those potential mediators in this risk to physical mental health comorbidity pathways. What it looks like so far is like this association is probably a little bit more complex than we expected. So there might be a very time-specific element here. And what's also possible is that something we observe across a range of research studies is that it's not necessarily single methylation sites that might mediate this association, but more like global composite methylation risk scores. So those could be risk scores for inflammation or methylation risk scores for cortisol or for epigenetic aging, that really mediate the link between gene environmental risk and physical mental health comorbidities.
Perfect. So you're trying to connect all of the dots, right? Your mental health, your physical health, through genetics, through epigenetics, and my listeners should be hopefully familiar with those methylation risk scores. I've chatted about those before with Dr. Michael Thompson. So yeah, and is it then true to say, Dr. Walton, that you've created then a brain age, or and have you created an epigenetic clock for the brain age? And can you tell me a little bit more about that?
Well, we used one. We haven't created it ourselves in our lab.
One.
It's not an epigenetic brain age, although there is a marker that we have studied in our research, and I can maybe explain that study in more detail. But the brain age measure we're using is based on brain morphometry data, so structural brain data, and it works similar to those epigenetic age clocks you talked about. So as you probably know, there are like a whole range of different epigenetic age markers. There's the Harvard clock and the Dunedin Pace, etc. And in the same way, there are a range of different brain age clocks. In a way, they can be sometimes based on structural measures such as the cortical thickness, or how thick your cortex is, or your surface area, or maybe volumetric measures that could be based on the white matter amount in your brain, so how different brain regions are connected, or functional connectivity. So there are a whole range of different brain age measures, in the same way how there are a whole range of different epigenetic age measures, and they're all tag slightly different concepts. So the brain age measure we use is based on a combination of, I think, around 100 different markers of cortical thickness, surface area, and subcortical volume. And in a same way as epigenetic age, it's a biological age measure of your brain. So it tells you kind of how old your brain is, and then you can calculate the discrepancy to your chronological age. And you can see how that predicts certain health outcomes. So, for example, just in the context of brain aging without epigenetic age, we saw that patients with schizophrenia, for example, show an advanced brain age compared to healthy controls. Now, what we were interested in is to see if that measure of brain age is independent of all of those measures of epigenetic aging you can obtain. And we looked at a population-based sample in young adulthood. So those were individuals between the age of 17 to 24. And we had measures on epigenetic aging. So we had the Harvard clock, we had PhenoAge, we had the Dunedin Pace. And we also had, that's quite interesting, a cortical, as an epigenetic age measure of cortical tissues, but obtained from blood samples. So you can use blood samples to get at a cortical epigenetic clock of brain aging, in a way. And then we correlated all of this with our structural measure of brain age. And the super interesting thing, as we found, is that they're actually quite independent of each other.
Oh, really?
Yes.
Yeah. I would guess the opposite. My hypothesis is that they would be not perfectly correlated, but that you would see a pretty strong correlation or association.
Yeah, and we didn't, and we were trying to make sense of that for a while. And one of my students who was leading on that project, she coined that term, or she found it somewhere, the mosaic of aging, that, it's well possible, right, that we have different ages, right, that some of our organs just age at a different rate than others. So the age we get at when we look at blood-based methylation markers is different to our brain age, also different to our cortical epigenetic-based clock. And it's possible that these are largely independent, maybe during early development, but as we go through life and as we adopt certain lifestyle factors and as we generally age, as we get older, maybe those different age markers become more and more interlinked to influence then, motor morbidity, coming back to that intersection between both physical and mental poor health.
I'm curious to follow along that data. That's definitely a surprising finding, to put it quite simply. I'm curious about the Dunedin Pace in particular in the correlation of brain age, because I know in the creation of the Dunedin Pace algorithm, they, well, let me back up and say people who have a faster Dunedin pace actually have less cortical thickness, they have less surface area of the brain, and they have differences in that white matter hyperintensity based region. So yeah, that's very interesting. Even if we're getting those measurements that are included in that brain age with the structural data,
Yeah.
how we see those differences. So
I, I
yeah,
mean, it's possible that it's just that maybe those associations emerge at a different developmental window, and maybe they
correct.
also work in combination. So maybe those measures, even though they're largely independent, maybe during adolescence, we've seen in other studies that in
Mm-hmm.
combination they predict functional outcomes such as cognitive decline actually much better than if you just looked at an individual epigenetic age or brain age measures. So, each one
Mm-hmm.
contributes a little bit to the overall prediction of the model, again suggesting there is probably a link between those more physiological epigenetic age markers and the more brain-based brain age
Right.
markers.
We need to look at everything together. I think another point, as well, the preprint actually just came out like a couple weeks ago with Raghav Segal's paper. He's out of Yale, where he has created those systems aging clocks. And
Yes.
yeah, if you've seen those,
Yeah,
yeah.
I
So
have.
I'm sure what he used for, I'm 90% sure there's a brain clock. I'll have to go back and check. It may not be one that actually made the cut. But if he does, that would be interesting, because I bet what he did to create the brain one is take a lot of that structural brain data. So I bet that one is probably going to be the one out of any of them in terms of the epigenetic clocks that would have the best or highest correlation with your brain age. So I'd be curious to see the connection there. Mm-hmm.
Really good point. Yeah.
Yeah. And kind of moving on again, you're doing great with this flow of conversation, Dr. Walton. I'm having a great time chatting with you. You mentioned previously developmental timing. So yeah, tell me a little bit about that. Maybe let's first define developmental timing, and then why is that important when we're looking at all of this stuff? Because you mentioned the reason that we're seeing these differences might be due to developmental timing.
So the developmental timing, I suppose, is a bit of a complex issue, but it
Yeah.
fundamentally refers to the fact, or it combines different findings we observe. So first of all, that really weird findings that we're more likely to identify longitudinal compared to cross-sectional findings or associations, and that there might be a certain developmental windows for vulnerability, for example. So to maybe make this a little bit more concrete to describe what I mean with that, we did a study on ADHD, so an epigenomite association study on ADHD, and we did it twice now, and twice we observed quite similar findings. So the study design was both cross-sectional and longitudinal. So we had ADHD symptoms in childhood measured in children around age 7 to 10. And then we had DNA methylation data either obtained at the same time point, so cross-sectionally, the same time point as ADHD symptoms, or longitudinal at birth already, so about 10 years earlier. Now, intuitively, I suppose you would think that when you look cross-sectionally, you're more likely to detect those associations, just because if two events are closely measured next to each other, they're likely to be connected.
Mm-hmm.
But we observed the opposite. So we didn't actually identify any cross-sectional associations. And that was a meta-analysis. So that was a quite robust finding across a whole range of different data sets. But instead, we found longitudinal associations that DNA methylation at birth predicted later ADHD symptoms over like a 10-year time period.
Mm-hmm.
And that's quite surprising, right? Because you wouldn't necessarily expect, those long-term predictive abilities of epigenetics at birth, predicting symptoms in childhood. But this is something we observe across a whole range of studies. So we did a study on measuring the ventricles in the brain. So this is this area deep within your brain. It's like this cavity. It's filled with fluid, and it has been linked to Alzheimer's disease. It has also been linked to schizophrenia. So anyway, there's this region in your brain And we tried to predict that using DNA methylation data. And again, just like with the ADHD study, what we found is that when you weigh your methylation risk score by information obtained at birth, you get better predictions, even if the brain measure you're analyzing was obtained, 10 or 20 years later. So that was quite surprising, quite exciting. So this is kind of what we mean with developmental timing. Just like, watering those plants, you don't observe it at the same time, but, if you wait a month, if you over or underwatered your plants.
Yeah. No, exactly right. So you touch a little bit on why it's important to look at these relationships between DNA methylation, early life stress, the brain, and the mental health. So it all comes back to this preventative based approach, right? By being proactive, by, knowing if you're overwatering or underwatering the plant when you're doing it, rather than, waiting and seeing what happens. So the DNA methylation and looking and analyzing at these relationships give us, I guess, insight into later health or future health state. Is there anything else you want to add there about why it's so important to look at all of these connections?
Well, I suppose it's also important not just in relationship to future health outcomes, but also to in relationship to risk exposure. So you could think that risk exposure during certain developmental time periods might have a larger impact on your epigenome or maybe on your personal health than had you been exposed to that same risk or trauma at different time points. And you could have, in a way, a whole range of different hypotheses that you might want to contrast. So, you might say, actually, the earlier you are exposed to trauma, the more severe the impact is, or maybe there are critical windows, or maybe it's just a dose response effect. So the more frequently you are exposed to adversity, the larger the impact on your health. So you can see those are different developmental theories, and they would lead to different predictions and different prevention and intervention programs. So what we did with collaborators in Boston, so this is lab of Aaron Dunn, and they're leading there, and we're involved in that. We developed this statistical framework. It's a method. It's called SLIGMA, Structured Life Course Modeling Approach, where we can directly contrast those different developmental theories. So does risk exposure at specific developmental windows lead to increased risk or a larger impact on your DNA methylation compared to other time points? And this is exactly what we find. We find that, for example, the study we just published on, which we could also replicate, is that risk exposure during the years between 3 and 5 years of age seem to impact DNA methylation later on more strongly so than risk exposure at different developmental periods. And that's quite important, right, when it comes to prevention and intervention programs. Can we really identify those critical windows? Of course, we're still long removed from actual interventions, but if those findings, replicate over and over again, we know better who to target and who to support best.
Yeah. And when you, just to be a little bit more specific there as well, when you're saying that you're seeing these DNA methylation changes later on in life when it's between a certain developmental window, what do you mean by DNA methylation changes? Or is that like with another EWAS study, or are you seeing things from like more of the biological aging clock or different acceleration levels? Can you go a little bit more detail into that?
Yeah, sure. So that was generally an epigenetic, like an EWAS framework, where we look at individual epigenetic markers, so different CPG sites. So we didn't look at epigenetic aging there. And
Perfect.
So that was DNA methylation wide data obtained at age 7. And then we checked again at age 15, so different time points.
Yeah, that's something that I really struggle with in explaining to people as well, right? When I think there are a couple studies out there looking at different, ACE-related events and, adversity events and traumas when even there's a couple of studies in a cohort of 13-year-olds and then another one in 18-year-olds with the Dunedin Pace and, how those traumas affect the pace of aging and some other epigenetic-based clocks. But I think that's really hard because the older we become, it's almost like we forget, or we don't know if we underwent some type of trauma, right? So that can be very difficult for people to dive back into. I find, at least when talking to people about reasons as to why maybe they have some accelerated biological aging. So I think, yeah, being able to have a predictor of that somehow that just looks at maybe those certain CPG sites or how those are differentiated, methylated, and look at them in more of a single analysis rather than these clocks could be very helpful.
I
Yeah.
agree. Yeah.
And okay, Dr. Watton, so you also state something I think pretty regularly as well in your work. You say, aging starts at birth, and that may seem, I guess, rather intuitive, but what do you mean when you actually say that?
Uh, it's interesting that you say this is intuitive. I mean, I think it's intuitive, right? But
Yeah.
if you ask people, if you just go down the street or something and you ask people what's aging or something, or they often think of old people, and you also see that in the research landscape, right? So if you Google aging and then you analyze the data sets that are being used in those aging studies, they often focus on people age, 40, 50, 60 or older. And so it's interesting, right? Because you think like, well, hold on, let's say you have a 30-year-old, is that person not aging? Or, and then you wind it back even more. Let's say you have a 10-year-old child or something, like, is that person not aging? Well, we would say like, oh, the child is still developing, it's not aging, it's developing. And you think like, well, is that the same or is that different? Is it just a different name for the same biological process? Or is there some kind of mirrored thing going on that development is the opposite of aging? So I was surprised to see that aging is only almost exclusively studied in the context of old age. And those research studies usually look at outcomes such as cognitive decline, or grip strength, or brain atrophy, and you can immediately see where the challenges lie, because now let's say we go back to that 10-year-old child. Can you really measure cognitive decline? It's tricky. Can you measure brain atrophy? That wouldn't work. Grip strength also isn't the right measure. So even though when we think about it and we say like, well, yeah, of course aging starts at birth, the next question then is like, well, how do we even study that? How do we study aging at birth? And I think that's where those epigenetic age measures and those brain age measures come in really well. I mean, in the area of epigenetic aging, we have some really decent measures that work really well at birth. So we have 3 epigenetic gestational age clocks. We have, pediatric epigenetic age measures, based on buccal samples. In the area of brain aging, it's a little, there's some measures out there, but I think there could be a little bit more diversity, and maybe that's coming with time. I mean, the area of epigenetics is really inspiring here, with the first, second, and third generation clocks. So I hope, we get there with brain aging too. And then I suppose the challenge is like, can we really measure aging from birth onwards? Can we predict somebody's life course aging trajectory? And then can we identify measures that help people age more healthily? And can we identify risk factors, but also, like, really importantly, protective factors? And when are those protective factors best placed? That's coming back to those developmental timing effects. So let's say engaging in exercise. Let's say I didn't exercise when I was a child and I didn't start jogging until I was like 30. Is that too late? Or is that still fine? Changing your diet or your sleep habits or something. At what age is it best placed to make a good impact on your general aging trajectory across the complete lifespan?
No, I mean, those are real questions. Those are practical questions. That is what everyone wants to know the answer to. I often struggle with that on my podcast. The reason I created this in the first place is to bring light to a lot of the research that we're doing. But people, this research is so important. I truthfully enjoy every single conversation I have with the people I basically, interview and have these chats with. But a lot of times people are like, okay, well, you tell me exactly what I need to do, right? What's best for my epigenetic methylation? What supplement can help? What medication? What can I be doing? And, I think we're getting there, but I think it's going to take time. I really do believe we're undergoing like an epigenetic revolution right now. And we'll only start to understand this a lot better and have a lot of those correlated based studies that are always going to sit at the framework of the research you're doing. So, we are going to have to create better predictors for people. You could actually argue, Dr. Walton, that aging starts at the inception, right?
Sure.
So, that's a whole different story. Like you were talking about those gestational age clocks, but you could argue that it starts at the inception and then, hey, what are you supposed to do from inception up until, 90, hundreds past that? What is the best at every single stage in your life from a lifestyle perspective or, some type of other interventional perspective? So,
so in the area of brain age, I suppose, what was really nice. So brain age, but also like generally just describing cortical alterations in people with mental health problems. So something we found, when it came to interventions is that, so we studied people with anorexia and looked at their structural brain alterations. We also looked at brain age. We roughly found similar findings. And what we found is that those structural brain alterations are actually not permanent. They can modify again with therapy success. So we looked at people, who just started therapy. So they were severely undermalnourished and just got into the hospital. Showed very high symptoms of depression and anxiety. And then we looked at people who were already undergoing recovery. So they already, had some weight gain, their symptom severity reduced a bit. And what we can see that was reflected in their brain structure. So we could see those changes, renormalizing again in a way. I mean, renormalizing is a bit of a contentious term, but, we could see that those really extreme alterations we observe when people are severely ill can change again with therapy, for example. And I thought that finding really resonated with people. So I got contacted by people who thought like, oh, this is a fantastic finding because it shows that, if you engage in therapy or something, and if you seek help, you can make a difference. And some of those changes, if you intervene quite early on, might not be as permanent as some feared. So, there's really some hope there.
Yeah, I think that's extremely motivating and hopeful for, again, we talk often about from an epigenetic standpoint, you're more in the driver's seat. Like you mentioned at the beginning, 30% of kind of the brain functionality or however you'd like to put it is due to those genetics. So there are ways that we can modify this through the exposome or again, different environmental factors. So what about, you just kind of hinted on an application or a commercialization part of your testing is, hey, we can measure this, we can intervene with a therapy and see a positive outcome or change. Do you have any other ideas on application moving forward with some of your work? And if not, that's totally okay. I just want to ask.
Well, I think, in terms of individual applications and, personalized medicine, we're probably not quite there yet, to really make this work. I think those risk measures we're using some of them work really well on a group-based level or population-based level. And I think the main application there is really to understand risk pathways and, as I mentioned before, when can we intervene best? In terms of personalized things, I'm looking forward to see how the field is developing. I know in cancer research, for example, the field is much further in terms of pharmacoepigenetic treatments, in terms of disease progression prediction, for example. In the area of mental health, we're unfortunately not quite there yet, but maybe one day that would be cool.
Yeah. Yeah. And then, what about you personally? What's next in terms of your investigations or studies? Is it still kind of identifying the connection with DNA methylation and epigenetics? What are you looking at next?
Well, we're looking at a whole range of things.
Yeah.
Let me just think
I know that. Yeah, I know you're busy.
I think something I'm really interested in is, so we just started the study on GWAS on brain age to really understand the genetic architecture of
Got it.
brain age. And once we have those results, there's a plethora of follow-up studies you could do. So, for example, you could see how the genetics of brain age overlaps with the genetics of epigenetic age. Is it generally a shared genetic, overlapping genetic architecture, or are they like individual components? And then once we did that study, we can also see use more causal inference methods. There's this really cool method called Mendelian randomization, where you look at genetic markers as proxies to answer quite causal questions. And that would allow you to assess, for example, if epigenetic age is a causal predictor of brain aging, or maybe a downstream consequence, or maybe they're not causally related at all. And I think those would be fantastic follow-up studies. I'm really looking forward to those.
Yeah, I think so. You're definitely gonna. I'll keep an eye open for those studies, but you'll definitely have to keep me updated. So, no, this has been great, Dr. Walton. I have a couple more questions left here for you. I want to talk about the methylation, imaging, and neurodevelopment, the MIND program that you've set up, or this consortium. And yeah, tell me a little bit about that and what you all are doing, what you're trying to understand.
Sure. So the MIND consortium was set up by a colleague of mine, Professor Cecil in Erasmus MC, and myself here, and we're co-leading it together to really study the associations between DNA methylation and the brain in a developmental context. So it was set up because there's not a lot of data out there in that area. So we're looking at data to understand the association between DNA methylation and the brain in early developmental periods. So from birth onwards, maybe even prenatally, if you like. But the studies that have data available are often quite small. So maybe 50 individuals, 50 newborn babies or something. And right, in epigenetic research, you need really well-powered studies, ideally thousands or 10,000s or even more to identify those really robust associations. So what we did is we founded this MIND consortium to collaborate with researchers and research groups across the world who have data available, like repeated measures data available of DNA methylation. So it's different time points, and of brain imaging at different time points, especially in the very early developmental periods from birth onwards. And we did that to really study the complexity of those really early life associations in more detail, to really assess the associations between peripheral-based, often blood-based methylation and brain-based processes to, understand those early developmental tricks. Because we know, right, that mental health conditions often have a quite early neurodevelopmental component to it. So we have, disorders like autism or ADHD, and even disorders like schizophrenia, there's this idea that there's a very strong neurodevelopmental component to it. And most people who develop mental health conditions develop them quite early on before they turn 18. So we really need to broaden out this early developmental time period and collaborate importantly together, so really pool data sets, work together, and really identify those very robust associations with more and better data, ideally longitudinal in early life.
Of course. It's really a way to see what everyone else is doing in the space and how we can all collaborate together. So I'm looking forward to how that consortium actually helps advance the field of neuroimaging epigenetics. I'll be, like I said, excited to follow along. So this has been great, Dr. Walton. I really can't thank you enough. For anyone else who wants to reach out or find you, how can they do that? I know you're active on Twitter. I think I found you on Twitter initially, alongside with some of your research, of course.
Yes, I'm on Twitter. I think my username is Esther Walton 18. So you can find me there, or you can just look up my webpage at the University of Bath in the UK, where you find my email address, and then people can just contact me through email or Twitter if they like.
Great, great. I'll make sure to include those links in the description and on my website and whatnot. Dr. Walton, my very, very last question for everyone that I ask is definitely has nothing to do with your research, but if you could be any animal in the world, what would you be and why?
Any animal in the world. So many
Anyone
animals.
you want.
Dead or alive, like dinosaurs and stuff.
I don't think anyone's ever picked a dinosaur, but it's not off the table. Yeah, I don't see why not.
Hold on. Well, I mean, dinosaurs is tempting, but let me think about this for a moment.
Yes.
No, I'm not going to pick dinosaurs, although it is tempting. It is tempting. So there's a running joke in my family. So the kids make fun of me because, well, I totally disagree with that statement, but they say I don't have any other hobbies but work. And I would like to say for the record, that's not true. But to prove a point, they did in best scientific manner a little survey at home to ask around. And for some reason, it turned into a survey about favorite animals, and I didn't have one. And so they assigned one to me, and they assigned the zebra as my favorite animal. And I fully embrace the idea of being a zebra, being all, white and black stripy in the savannah, minding my own business and hoping to escape the odd lion along the way. So I take zebras.
I like it. No one has said that before, so we'll go with the zebra. That's amazing. Thank you so much for answering that question, and I like the little backstory behind it as well. So you can send this recording to the kids, and now you have it on record that you're not always working. You're having fun talking about your research too.
Thank you so much.
Well, thank you so much, Dr. Walton. Thank you, everyone else, for joining us here at Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn how. Thanks again.
Bye.